Hi, this is Steve Swanson. I'm head of Amgen's Clinical Immunology Department, and we're going to summarize what you've learned in Immunogenicity 101, the immunogenicity of therapeutic proteins. So the objectives of this portion really to review and recap some of the important concepts regarding immunogenicity of therapeutic proteins, to review some of the unique characteristics of selected patient populations as related to immunogenicity, and to review the features that differentiate an early immune response from a more mature immune response. As we think about the antibody response to therapeutic proteins, it's important to recognize that the induction of immune response is a very complex process. Contributors include the product quality attributes, the disease state of the patient, and the status of the patient's immune system. Historically, many therapeutic proteins have induced antibody formation. The effects of this can range from minimal to significant. Some of these are associated with severe adverse events. Both the pharmaceutical industry and regulatory agencies continue to partner for a better approach to immunogenicity assessment. So really one of the things that's very important for us to recognize is that the immunogenicity of a therapeutic protein is something that we can't predict reliably. We have to establish this during clinical trials. We have to collect as much data as we possibly can to really understand what's happening with our therapeutic proteins. And while many times we have a fairly good feel for what the immunogenicity of a therapeutic protein is going to be during our clinical development phases, things can change when the drug is in the marketplace, and we have to be responsive to that. So it's important for us as an industry to really understand the life cycle management and understand that immunogenicity is something that can change as product quality attributes might change during manufacturing, and we have to be responsive to those changes as we continue to develop these therapeutic proteins.